Bismillahirrahmanirrahim, and welcome back to this next video. And this is the 11th video of the chapter 2 of Immunology. And from the last couple of videos, we are talking about the granules in the neutrophils. And in the last video, I started on the secondary granules, which is a specific type of the granules that is there in the neutrophils. Now, I told you that these secondary granules, they are made when the neutrophils, they move from the myelocyte to the metamyelocytes. And then I gave you an important example of the antimicrobial component, an antimicrobial protein or protease that is present in the secondary granules, and that was the lactoferrin. And we had a detailed discussion on the lactoferrin, that the lactoferrin is an iron binding glycoprotein, and it is actually responsible for the sequestering of the iron, and this iron is very important for a variety of the functions of the pathogens like the energy metabolism, the DNA synthesis, the virulence factors, and then I gave you a lot of examples of the virulence factors that are there in the pathogenic organisms and how the lactoferrin can actually stop their function. Now, in this particular video, I want to focus on another important antimicrobial component which is present in the secondary granules, and that is known as the collagenase. When you talk about these collagenases, this is actually a class of the enzymes known as the metalloproteinases. We call them as the metalloproteinases because they need a metal ion for their activity, and it is specifically belonging to the matrix metalloproteinase family. When you look at the human body, you are going to find a lot of the metalloproteinases, but the one that are involved in the degradation or acting on the matrix proteins, they are known as the matrix metalloproteinases. So this collagenase, it actually belongs to this family of the matrix metalloproteinase. Now, these enzymes, they are involved in the breakdown of various components of the extracellular matrix, as I've told you, including the collagen. And when you look at the collagenase that is secreted by the neutrophils, or you can say the one that is present in the secondary granules of the neutrophils, they are known as the neutrophil collagenase. Of course, it does make sense because this collagenase is present in the neutrophil, and sometimes they are also known as the collagenase 1. Now, these collagenases, as I've told you, they are responsible for the breakdown of the collagen. So if you look at the structure of this collagen is a fibrous protein with a unique triple helical structure, and it consists of 3 polypeptide chains. Now, that means you're talking about a stronger protein, because if you are getting a triple helical structure, that means that the collagen, they are very important for providing stability to the cells and in the extracellular matrix. Now, the collagenase enzyme, what they do is that they recognize specific amino acid sequences within the collagen molecule. Because this collagen is a very large molecule, there should be something specific that the collagenase is going to recognize. And what these collagenase enzyme do, they recognize specific amino acid sequence within the collagen molecules. So, what are those specific amino acid sequences? If you look at the amino acid sequence that the collagenase recognize, so the collagenases, they are actually going to recognize the peptide bond between the glycine and a bulkier amino acids, and that is typically the phenylalanine or leucine in the collagen triple helix. So what that means is that when there is a peptide bond between the glycine or the phenylalanine, or if there is a bond between the glycine or the leucine, the collagenase is very good at recognize those particular peptide bonds and thereby breaking them down. Now, these collagenases, as I've told you, this is a metalloproteinase, and in this particular case, they require a metal ion, which is particularly the zinc ion. So the zinc ions, they are very important for the function of the collagenase, because the zinc ion, it is present in the catalytic side of the collagenase enzyme. So the zinc ions, they are very important and they play a crucial role in the catalytic mechanism of the collagenases. Now these zinc ion, what they do is that the zinc ions within the active site of the collagenase, or you can say the catalytic site of the collagenase, they are going to help coordinate and stabilize the substrate, facilitating the cleavage of the peptide bond. So what the zinc ion is doing, it is making a stronger interaction between the collagenase and the collagen, and once that particular interaction is stabilized, the catalytic site can actually go for the breakdown of the peptide bond, whether that is between the glycine or the phenylalanine, or that is between the glycine or the leucine, for example. Now what the zinc ion, it helps polarize the peptide bond, making it more susceptible to a nucleophilic attack, because you are interested in the breakdown of the collagen, you are interested in the breakdown of the peptide bond between the glycine or the phenylalanine, for example. So what the zinc ion do is it is going to polarize this peptide bond, making it more susceptible to a nucleophilic attack. So I'm not going into the detail what a nucleophilic attack means, but simply when you talk about the nucleophilic attack is when an electron-rich species, which is known as the nucleophile, that attack an electron-deficient species, which is known as the electrophile. Now, in the context of the neutrophils, when you talk about the neutrophils, so the release of this collagenase is actually part of the immune response to infection or injury. Now, what you do is that this collagenase help the neutrophils navigate through the extracellular matrix, allowing them to reach to the site of infection more efficiently. As I've told you that these collagens, they are very important protein in the extracellular matrix. They are very important to provide stability. So if they are there in their stable form, the neutrophil, they will find it very hard to reach to the site of infection. But when the neutrophil, they release these collagenases, the collagen, they are broken down, the neutrophil are actually going to find a way to enter into the site of infection and then perform their immunological function. So this is very much logical why the neutrophils, they are releasing these collagenases. Now, overall, the activity of the collagenase is tightly regulated in normal physiological condition, because if the collagenases, they are released all of the times, they degrade the collagen all of the time, that is going to affect the normal physiological functions of the cell. Because if they are not stable, if the cells, they are not stable, they are not actively binded to their extracellular matrix, a lot of normal physiological functions, they are going to get affected. So the activity of the collagenase is tightly regulated. They are only released when they are needed. For example, if there is an infection. Because if you talk about the regulation, so dysregulation of the collagenase activity, that has been implicated in various pathological conditions. For example, the chronic inflammatory diseases, the tissue degradation like the arthritis, and in certain cancers. So that means you have to tightly regulate the release of the collagenases so that the collagen remain in the stable form and it perform its normal physiological functions. Now, if you look at this one, these three lines, these three helices, that is actually the structure of the collagen. If this is, say, for example, the active site, and these are the helping domains. So what they do is, if you can see in the this B over here, these active site, they are actually going to pick one of the helices, because it's very difficult to cleave all of the helices. So what they do is they are going to pick one of the helices, and when the helices, they are broken down, you can actually see the collagen that is going to get broken down, and the neutrophils, they will find it easy to enter into the site of infection and perform their immunological functions. The third important antimicrobial component that is present in the secondary granules, that is known as the lysozymes. Now these lysozymes are also present in the primary granules, which are also known as the Azurophilic granules, and over there I had a detailed discussion how these lysozymes, they are going to work. But here I'm only going to give you an overview of that. So if you are interested in detail, I'll share the link in the description of that particular video, and you can see the details over there. Now the lysozyme, these are enzymes which are present in a lot of body fluids like the tears, the saliva, the mucus, and of course we are discussing about the neutrophils. They are also present in the secondary granules and in the primary granules of the neutrophils. Now these lysozymes, they prevent against the bacterial infections, and what they do is they are going to recognize the peptidoglycan layer of the bacteria, and once that is recognized, it is going for the hydrolysis of the beta-1,4 glycosidic bond. As you can see over here, this is actually the beta-1,4 glycosidic bond, and what these lysozymes do, they are going to break it down, and when that is broken down, the peptidoglycan layer of the bacteria that is destroyed, and the bacteria that is going to get killed. So if you like the video, please subscribe to my channel, hit the notification bell. If there is any question, you can ask me in the comment section.